IVM.
Hello and welcome to the Pragati Podcast. I am not your regular host, Pavan Srinath. Instead, I am a recurring guest, Anupam Manur, who has now usurped Pavan's position as the host for this particular podcast. However, if you're going to miss Pavan, I do have Pavan with me as the guest. And what we're going to do today is talk about medicines and the whole industry and the process of discovering new medicines in India and worldwide. Now, pharmaceuticals in India, for example, has been of great importance in the recent past. It has earned us foreign exchange. But one of the crucial questions that we have to consider is whether India is just good at copying medicines and producing generics, or are we developing our own medicines as well? Which then would normally lead to the next question: what does developing a new medicine or a drug actually entail? To talk about all of this and more, I have with me Pavan Srinath. Welcome to the Pragati Podcast, a weekly talk show on public policy, economics and international relations. I'm Anupam Manur, resident economist at the Takshashila Instituion and a wannabe podcast host. We'll start a conversation with Pavan in the guest seat after a short break.
Hi everybody, welcome to another awesome week on the IVM Podcast Network. If you're not following us on social media, please make sure you do. We're IVM Podcast on Twitter, Facebook and Instagram. I also kind of wanted to make a couple of other announcements, right? So first, as I think I mentioned last week, I just want to kind of reiterate, we're going to be closing our SoundCloud channel at the end of this month. So if this is where you're hearing us, please maybe try and listen to us on something else, maybe the IBM Podcast app, maybe another app. We're on all kinds of different platforms. The other thing is a little happier, right? So for the first time ever in Asia, we have a podcasting award for shows made in Asia. And we're very happy to announce that a number of our shows have been nominated. Pesa Vesa, Tech Careers in the New, Varta Lab, and Junior One have all been nominated for these awards. Please go to the website asianpodcastawards.com and go vote for our shows. Come on, make us win, guys. On Cyrus Says this week, check out the episode we did with the cast of The Upstarts, the new Netflix series. On the Empowering series, Zarina is joined by stand-up comedian Shrija Chaturvedi. On this episode, Shrija talks about her journey as a stand-up comedian, the way she perceives her struggles, and what makes her feel the most empowered. You thought Simplified were done with celebrating their 150th episode? Not even close. Chuck, Naren, and Shriketh are back with the third part of their 150th episode. Make sure to tune in to see if your questions are featured. On KBCD, Farhad and Sunetru talk about the evolution through college life and their experiences that changed them forever. This week on Boundless, Natasha reads the poem calling out all aunties who pressurize us to be a certain way and shares her idea of a charming prince. On Golgappa, host Trupti is joined by actor and contestant on Big Boss Marathi, Neha Shitole. She talks about her experience in the Big Boss house and the politics in it. On Feeding 10 Billion, Ramya and Varun talk to food historian Kurush Dalal. They delve into the history of our food and how it dictates our future. On Agla Station Adulthood, Ayushi and Ritasha talk about celebrating girlhood on Girl Child Day. Together they talk about the situation of women and girls in the country and how important it is for them to have powerful role models, a sense of freedom, and the ability to exercise agency. On The Habit Coach, Ashton talks about daydreaming and how one must engineer their imagination. With that, let's get you onto your show.
Welcome back. Hi, Pavan.
Hi Anupam, this is a fun role reversal.
Yeah, I'm not really used to this, but I've been with you enough number of times to hopefully get some of the good...
aspiring podcast host, I mean, you should have started one already by now.
Yeah,
but I'm glad you are at least coming back very often to the Pragati Podcast to talk about various things.
Right, so let's get down to the topic.
Yes.
We are talking about medicines in India broadly. Healthcare by itself is a massive topic. We are beginning to at least think about healthcare. You'd in fact done a study on how much India spends on healthcare, and until then we didn't even have a clue as to how much we spent on healthcare. And now we're talking about Ayushman Bharat, for example, a scheme, though it's in its infancy at best, about providing healthcare insurance and coverage to the whole of India. So this topic is at least now in conversation on one side of it. On the other side, we're actually beginning to talk about medicine as well. We have an entire national list of essential medicines for which we try and control the prices. There's an entire conversation going on, generics in India, IP laws, a whole set of things. But before we go on to such big topics and, which requires a lot more detailed exposition, let's talk about medicines in India. Right? Now, there are for example, a whole set of diseases which are sort of peculiar to India, which you might not find in the rest of the world. If you could take, for example, just mosquito-borne diseases. Now, since it's been raining in Bangalore for the last 3 or 4 days, I'm sure that will become a problem in the coming days. So let's just pick up on that. And you have at least 3 or 4 variants in the recent past that has come about. You have dengue, you have chikungunya, and of course malaria, which has been around for the longest time. Now, while you have some form of medicine for malaria, you don't really have a lot of medicines for chikungunya and dengue. So how or why is that?
Thanks, Anupam. So to start off with, what most of us living in Indian cities really face and dread, right? One is a whole host of, lifestyle and sort of genetic challenges that we face with, from diabetes to heart disease and so on, and everything associated even with a little bit of age, and all the infectious diseases that India is yet to conquer, right? So even malaria was something that we were aggressively attacking between the 50s and the 80s, and we actually declared victory against malaria, even though while we had dropped the rate of malaria down to a fraction of what it was half a century ago. We then sort of slacked off. Things like dengue and chikungunya, we don't know how to address properly. And first of all, over here, when we are talking about viral diseases, the number of antiviral drugs that are available and that have ever been invented are handful.
Right.
In fact, you have more vaccines against viruses than you have actual drugs. Most of the drugs made against viruses are for HIV. Which is a complex beast in itself, because HIV is something that mutates rapidly. You give it a drug, you don't take the second dose of that drug, HIV might have become resistant to that drug already.
Right.
It's quite insane. Outside of HIV, we only have 2 or 3 antiviral drugs that have ever been created. There's, I think, Tamiflu, which is talked about when H1N1 was doing the rounds, and that's about it, right? So viruses are that much of an unsolved problem. The most common anti-infectious drugs that we know of are antibiotics.
Right.
And though antibiotics have come to mean this catch-all term, antibiotic is specifically a medicine that is antibacterial. Okay, and maybe
as the name suggests.
Yeah. So it's antibiotic, right? I mean, but if you actually look at it, bacteria do look like living things. Mm-hmm. If you actually see viruses, especially electron microscopy images and so on, they look like alien pods from somewhere, right? I mean, they have these crystalline structures, this hexagonal head, and weird body and some legs.
8 legs. And
yeah. So they look alien, and honestly, viruses by themselves are barely living. It's like a bit of code and a bit of hardware somewhere. They actually hijack our human cells or mammalian cells in order to use our human cellular systems to propagate, to create all the problems, to do other things. So fundamentally there too. So I don't even want to go near viruses today, right? While we want to talk about medicines for India. So the challenge really is that there has been a long neglected list of tropical diseases which the Western world never faced as much of, and as a result they were always neglected from drug development points of view. But funnily enough, if we talk about mosquitoes, if we go back to malaria, some of the biggest innovations in malaria also came from the Americans. Because Americans had their troops from all over the world going back to America after the war and picking up malaria as they went. And so they would, so long as the mosquitoes were there in certain parts of America, especially Florida and Southern America, these returning soldiers would introduce malaria protozoan and the disease-causing agent to the local mosquitoes. Right? So the Centers for Disease Control, the CDC, which we know today as this, reputed medical institution that does amazing work, tackling Ebola to managing other things, they were developed as a response to malaria.
Right.
Okay. So there has been interesting equations with the Western world on this, but by and large in Africa and India, and there are other places where people are poor, their ability to pay for a brand new medicine that's under patent is very hard, and therefore nobody wants to invest in that research. And right, so eventually I want to circle back to this idea of why we need to make some of these drugs, because no one else will make it for us.
But if is our best bet to have a US Army contingent get dengue?
You know what? That's actually not too far from reality. The reality is, you've heard of Zika outbreaks that are happening in southern parts of the USA, right? Why is that happening? One part climate change. One part is just that you have new viruses that are occurring. The range at which mosquitoes operate is expanding ever northward. And Zika and dengue are Mexican problems as well as American problems now, apart from ours, right? Which is why the one dengue vaccine that is in the market right now, it's called Dengavaxia. It's not yet been introduced into India. Lots of problems with it. It was developed by and large in the US and Mexico. Right? And we might have been sort of incidental beneficiaries of that. But, the dengue virus has 4 subtypes. What operates in India may not operate in Mexico. So you have all these challenges because diseases are increasingly extremely complex. And unless you've done enough research in your area, you won't be able to find an effective cure. And we are still talking about 20th century medicine, right? If we're talking about 21st century medicine like gene therapy, all these new fancy things, you need to study Indian genetics even more, right? So what you do in India will matter a lot more.
It's a whole new can of worms or
yes,
can of mosquitoes. Let's leave that aside for
yes.
now. Okay, now let's get back. Under the guise of trying to simplify it for our listeners, it's actually my own ignorance. And I'm going to ask you this question, which is, essentially, malaria, dengue and chikungunya, are they viruses which transmit through the mosquito into the human bloodstream? Is that how it works?
Dengue and chikungunya, yes. Malaria is by something called a protozoan. Right?
Okay.
So, if we go back to high school biology, there are the prokaryotes and the eukaryotes. And basically, you're bacteria and you have animal-like cells, right? So, single-celled animals are protozoans.
Right.
So amoeba is a protozoan. So there'll be a nucleus. The bacteria will just have DNA. It won't have a nucleus. So that's the difference. So the moment you get to a protozoan, you will have, your mitochondria, your DNA, all the things that we talk about in a human cell existing inside. Evolutionarily, it's a slightly more evolved organism. So it's Plasmodium vivax, Plasmodium falciparum, a few strains of that protozoan which transmit malaria. Right? And which is why we do have drugs against malaria. But we have none against dengue and chikungunya.
Interesting.
And not just that, we don't know what other things. Many other times you go to the doctor now, you will be told that you have a dengue-like fever, right? Or viral fever.
Yeah.
The reality is most viruses, we don't even know what is infecting most Indians here.
That was going to be my next question, my next stupid question, rather, that a lot of times we have flu, and that is broadly, you go to a doctor, it's classified as one of two. One is a bacterial kind of flu and one is a viral flu. Right. And the symptoms kind of change, whether it's high temperature in the evening, etc. But by and large, in my own experience, for example, you're given something to control the symptoms. So basic crocin or paracetamol so you take it when you have high temperature, but really no cure as such for the viral aspect of it. Whereas, if it's a bacterial one, you get antibiotics.
That's true. But even here, right, the word like flu, influenza is something which is a seasonal disease in the US. But influenza, so that is H5N1 or something like that, right? So influenza is a particular virus that might exist in India also, but we don't know as much about it. I personally found that taking an influenza shot, a flu shot vaccine, which is very seasonal and has to be changed every year or two, it seemed to help me in India, reduce the amount of times I fell sick. At the same time, the cold virus, the rhinovirus, is a different virus. And then associated with all of it is our throat infection is called the strep throat, because you have the streptococcus bacteria, which sort of rides on these viral infections and makes your throat swell up and, cough and phlegm to develop and so on. The antibiotic will only be against the bacteria. And there's this old joke that, without medicines, a cold takes a week to go away, and with medicines, it takes 7 days. Right? So with viruses, you manage symptoms, you treat the body well, eventually the body manages the infection, right? So, but in all of this, I think it's important for us to try and understand what does it really take for, a germ of an idea to become a potential new drug to something that you can buy off the counter or with a prescription at a medical store.
Yeah,
House,
exactly.
right?
So we do take antibiotics for a lot of the infections that you mentioned. And in fact, I mean, there's this whole debate of whether we are taking too many antibiotics or too little. And we'll come to that slightly later. That's something I'm personally interested in and very curious about. But even before we get there, how does an antibiotic even, that's what, as you said, go from, some form of testing stage to the counter? Even before that, could you just, explain to our listeners saying what exactly is an antibiotic? How does it work?
All right.
Is it that you're actually introducing bacteria so that your white blood cells begin to fight? I have no clue about these things.
All right.
So treat me as a fifth standard kid and explain how an antibiotic works.
All right.
And then how do you actually develop antibiotics?
Sure. So I'll flip that around because a lot of people, I'll slightly approach it from a historical perspective.
Sure, yeah.
Right? All of us have sort of grown up on this semi-mythological, semi-fairy tale version of how penicillin was discovered.
Yeah.
Right? So you had Alexander Fleming. I think he was working at St. Mary's Hospital in London, and he had this basement lab, and he was playing around with Staphylococcus aureus. So now, Staph aureus is this small round bacterium that's there, in the crack of your nose, on your skin, in armpits, pretty much all over the human body. And Staph aureus also, by the way, is the one which, when it develops resistance, becomes MRSA. Right? Okay. MRSA, which you would have heard of as this superbug, is methicillin-resistant Staphylococcus aureus. Right? So, penicillin-resistant bacteria.
Sure.
Right? That's all. And the story is that he was doing something and there was some fungus that grew on his plate because he had left it near an open window, and he comes to see that the fungus has actually killed the bacteria. Right? And that's the fabled story.
I hope you guys are not having breakfast or lunch at this point of time, but sure.
But it was this fungus that saved us from bacteria, right? But if you look at the history, there have been cases documented from even thousands of years ago of how moldy bread was used to treat wounds that would help in some way. Mm-hmm. Right? So it depended on certain kinds of mold, and basically those funguses secreted something, and therefore you got rid of the bacteria. So in the lead up to the 20th century, there were many reports, many people trying out that there is something with this particular fungus called penicillin.
Okay.
With it, something happens.
Right.
What Fleming managed to do was he managed to create a proper culture of penicillin. He was able to grow penicillin, the fungus, properly, and he was able to demonstrate that this thing worked against a range of bacteria. But, Fleming was famously bad at communicating. So Fleming does this in 1928, right? So this is before the war starts, 30s, we've talked about the GDP happening, coming into play, lots of things happening. And the war plays a huge role in the early development of antibiotics, right? So 28 or so, Fleming does this. After that, there are a few people who try out a penicillin sort of extract with a few patients. It works with few patients. It doesn't work with one or two. So they're trying to figure out what's happening. But one of the things that Fleming did not do, apart from document, right, all of that, is he did not test it on infected animals.
Okay.
Right? So he did demonstrate that, look, this seems to be safe, no one seems to be dying, nothing wrong seems to be happening, but he didn't really do that. So it really took two others. So that's why when you look at the penicillin story, the Nobel Prize for penicillin and related discoveries was given to three people: Alexander Fleming, Ernst Boris Chain, and Howard Walter Florey.
Right.
Now, Florey and Chain were, I think, Australian and British respectively. And in 1939 or 38 or so, they started new work.
So, whole 10 years after the initial discovery of penicillin.
Yeah, and apparently whenever Alexander Fleming published his stuff, most people didn't bother. No one was interested. And in the meantime, in the 30s, the sulfur drugs also started being created, right? And today sulfa drugs don't exist in the market anymore, by and large, but other antibiotic efforts were also taking place, right? So 39, they start doing some testing. They test extensively with animals, and then they start testing with humans. And it's funny that, when we talk about, there's a lot of biological revolution that is happening in how we study biology, the techniques we have, how much we understand. At the same time, we are developing amazing ability in synthetic chemistry, right? Okay. Because the war effort needed you to produce, so many different types of compounds, so creating, ammunition, weaponry, explosives, and so on, right? So you really needed to know your organic chemistry right. And as a result, you see this thing happening where they finally think that, look, there is something in penicillin, right? And there were lots of challenges because penicillin in the form that they made it lasted in your bloodstream for 4 hours.
Okay.
So if you have to give someone penicillin, you have to give them every 4 hours or every 8 hours. And so you need a lot of penicillin. In 1941, late 1941, early 1942, all the penicillin that was produced, and this is when Merck, the company, also gets involved in penicillin production. Merck, all the penicillin produced, half of it was used to treat one patient alone.
Okay.
The entire production of penicillin in the world at that time, half of it was used to treat one person.
Who's...
I forget the name, but there was one person treated. Okay. In 1942, there was enough penicillin made to treat 10 people.
Right. Okay.
Yeah. Because the penicillin, the synthetic method of making penicillin, purely from organic chemistry, was only discovered in 1961.
Okay. So before that, what would they do? They would actually...
So this was actually from you culture penicillium, the fungus, and then you grow it in certain media.
And then you bread out, wait for it to mold. And then
obviously not. But that's the broad idea. You create a culture, and then penicillin is secreted by this fungus when it's under stress. So you remove glucose at a certain point. So it thinks it's under stress. It releases those things out to protect itself. And, then you sort of extract it.
Just get them to Bangalore traffic, no? You'll have all the penicillin you want. Okay.
In all of this, this was also the early days of the predecessors of high fructose corn syrup. Right? People were using agricultural produce, tweaking up the sugar. So there was some such... sugar-heavy thing that was used as a medium. So they also had to find out the right penicillin strain that worked with that medium, etc., etc. And then there was another person who patented the process of making penicillin.
Right.
Penicillin itself never got patented, by the way.
Yeah.
And there were some people who considered it, and some senior scientists who advised them, don't patent it, it's for the greater good, all of that.
Fantastic, yeah.
Okay. And... with that, in 1944, I think, the production had ramped up to beyond 600 million units per year.
Okay.
And it had crossed 1 billion units by 1945.
Right, okay. And so how much is that enough for? I mean...
1 billion units as in... imagine you can treat 100 million people.
Okay, so significant.
So from 1 to 10 to... millions.
About millions in about 4 or 5 years, literally.
Yeah, less than that. 3 years.
Fantastic. Okay.
Right? So that's the kind of ramp up we are talking about when... And people say that if Fleming was actually listened to much earlier, maybe war deaths would have come down significantly. As it is, the development of penicillin and other drugs, people estimate that wartime casualties reduced by 12 to 17% in the World War. And the denominator
yes.
in the World War.
That is significant.
Right? Because of the development of antibiotics. And that is the effect that we've seen from ever since, right? But this already tells you, even in the early days where there were no extensive regulations or protections and you needed this for the wartime effort, you had this 19th century stuff that happened. Fleming does something in 28. From 1945 onwards, you actually start getting civilian use of penicillin.
Right.
So you can see that span right there in the early days.
Right.
Today, you might have better systems if you want, but you have a lot of regulation, a lot of safety checks, lots of things to go through, because along with the advent of penicillin and other antibiotics, you also had new safety concerns that cropped up later. People realized, you might have a drug that looks fantastic on paper, but there might be some secret side effect that's actually killing off many more people than you know. And that was the case with a few drugs much later.
Yeah, I mean, this was a very happy kind of story with the penicillin. It worked. There was very less side effects.
And I'm also sharing it shorn of all the messiness associated with it. Right? There's a lot of mess and a lot of scientific mess as well. I mean, you would have figured all of this out. Separately, at some point, the actual structure of penicillin was discovered. And then what it attacks was discovered. And that brings me to how antibiotics work, right? So penicillin is something that attacks something called peptidoglycans. These make up the cell wall of the bacterium.
Okay.
And these, so it goes and attacks this complex of a protein and a starch molecule, something like that, and goes and cuts that, and therefore the cell becomes weak. Water rushes into the cell. So all our cells have a delicate pressure, osmotic pressure, right, with salt concentrations, which is why you can never inject just pure water into your bloodstream. And as a result, so these cells sort of explode. So various antibiotics either make bacteria stupefied and sort of inactive, or they kill them. And they do this by attacking various parts of it. You can destroy the cell wall. You can destroy the cell membrane. Think of, trying to injure another person, right? These are almost like physical attacks.
Right.
And then you have poisons of various sorts. You will have essential enzymes, essential proteins that are crucial to the respiration of that bacterium, to the metabolism, how it does digestion. So you block any of those systems which are critical to the life of that bacterium, you can destroy that bacterium.
So what are these antibiotics by itself?
So they're small molecules.
They're
small organic molecules.
Okay.
So there'll be a benzene ring, there'll be some other ring, some CH3 groups here and there, some nitrogen here or there. The sulfur drugs have a sulfur atom working with some oxygen atoms. But these are largely small chemical molecules.
Sure. Now, we get that from your story that penicillin was found or discovered because of Fleming's, let's say, hygiene issues of leaving his plate out without cleaning it.
By the way, the plate was an agar plate of bacterium, not an eating plate.
Okay. Nonetheless, how do other antibiotics get discovered?
So after this, a lot of work was happening. So what you see is between 1949 or so, or 1947 and 1977, every year there were multiple antibiotics that were discovered. Many of these were variants of existing antibiotics. So you make it slightly more effective, you modify something there, something might have a slightly lesser side effect, something might just be more effective. Some like penicillin, which was naturally made, in a minority of people triggered allergic reactions. So you have something purer, less allergic profile, etc. Right? But the last in these antibiotics, some are considered broad spectrum, so they'll tackle a wide range of bacteria, and some are narrow. They'll only hit certain types of bacteria. And so even in our diagnostic process, when people are not sure what kind of infection you have, they'll hit you with a broad spectrum.
Oh, right. Okay.
Right?
Yeah.
And you can separately debate misuse, overuse, all of that. And only once they know, okay, you have paratyphoid. Okay, I have a specific drug that will work best against paratyphoid. Right? So that's when you can use a specific drug. And in this, I don't want to get into the details. In bacteria, you classify them as gram positive and gram negative. Basically based on how an old stain called the gram staining method, if the stain sticks to the bacteria, it's positive. It doesn't stick to the bacteria, negative, whatever. It's some way of classifying. For gram negative bacteria, which include a lot of important diseases, we haven't had a new antibiotic since 1962.
Right, okay. That's
and
a long time back. Yeah.
And like you earlier mentioned, with antibiotics, it is a cop and robber game, right? So there is resistance that is built. So you bring a knife, then somebody brings a gun to that knife fight, and then you bring a machine gun to that gunfight. Yeah. That is a process that happens. And in bacteria, it happens in two ways, right? So imagine there are 1 million bacteria. You hit that spot with antibiotics. It kills 99.9% of it. But there'll be a tiny part, 0.01% of those bacteria which naturally just have something right or wrong with them which makes them resistant to this antibiotic, and therefore only those survive, and then they multiply in no time to form the full population.
So use Harpic every day.
Yes. And even if you use Harpic, you actually are breeding these bacteria that are extremely resistant. The other way is actually they can bacteria have sex in certain ways. So they can actually transfer genes from one bacterium to the other.
Sure, okay.
So, and these are ways in which they develop resistance. So you need to constantly be creating new antibiotics. And while there have been some antibiotics that have been created since then, even in the 2000s, 2010s, contrary to popular belief, they're mostly modifications of older ones.
Right. Okay, now let's again take this. We are kind of speaking about discovery and how new antibiotics are discovered, how they're developed. And I just discovered that they're not developed as many as I used to normally think. Now, let's say once it's discovered, what happens then? Is that what kind of testing is done? As you said, it's not just that there is one type that you discover and start using it. You can have variants of it. You can modify that same thing. And for that, then, of course, you need a lot of testing, I'm assuming, right? And there's some form of regulations as to how many people you need to test. I'm not sure. Right. What is a minimum kind of sample size, etc. And then probably it'll get the approval of some regulator. Just talk us through that process.
Sure. So in this, honestly, even before we get to that regulatory process, the early form of drug discovery was like the Fleming story, especially the mythic Fleming story, right? The idea of serendipity and sort of common knowledge, if you will, leading to people isolating certain chemicals, certain compounds that work against a certain disease or something, right? So it was largely trial and error, serendipity, good fortune, chasing down leads. Beyond a point, there was a movement which got started where they talked about rational drug design.
Okay.
Because a lot of the times you would have this drug, you would figure out everything about it, you would know that it even works well, it even gets out in the market. You don't still quite know how the drug works.
Okay.
Even today, there is debate on how crocin or paracetamol really works.
You're kidding.
Not 100% sure.
And people keep popping crocin.
No, it works.
It works, but we don't know how it works.
I mean, there is a lot of debate on how it works.
Right,
it is very tough to measure sometimes.
Right.
There are certain pathways that are proposed, but particularly with paracetamol, not with painkillers, very tough to know how exactly it's working, right? So the rational drug design was this idea that, look, there are natural targets that are there in the body. Mm-hmm. Think of, in any cell, you have DNA. DNA encodes information. RNA is a temporary copy of that information. And that RNA is a code that is used to create a protein. And that protein acquires a three-dimensional structure and can do pretty much whatever you want it to, okay, right? And it's a large molecule. So these drugs are small compounds. These proteins are at least 100 to 1,000 times the size of a drug molecule. All right. So now think of these various proteins that are important as locks.
Okay.
And your drugs as keys. Right? So what you want really is to have a key that fits into the right lock and does not go fit into other wrong locks.
Sure.
So your side effects come from other wrong locks. Like with ibuprofen, think of it, once you take the drug in, you take ibuprofen, it goes into your stomach, it gets slowly absorbed in your stomach and intestine, enters the bloodstream, and then from the bloodstream, say you have an inflammation in your knee, you've fallen down, it will circulate all over your bloodstream, find that there's some stuff happening over there, and then it goes and inhibits the processes that cause your knee to swell up. So that's why it's actually called an anti-inflammatory, not an anti-pain, not directly, right? Yeah. Because it reduces the inflammation. It also
reduces the pain.
Exactly. Okay. Right? So now this drug has to do so many things, right? We need to know that there's some target there in our inflammatory processes like whenever there's an injury, the body hits panic button and then a bunch of things happen around there.
Sure.
Right? It knows that, okay, it has to go and attack something, but it has to be available everywhere before it. Rational drug design was this idea that, hey, if we are actually learning more about human biology and about disease biology, can we actually identify the targets that we are attacking? And then we know the shape of the lock. So can we do some lock picking and figure out what kind of a key we should get in there?
Yeah.
Right? You would have heard of the word enzyme.
Right.
An enzyme is a catalyst. So there is some reaction. Enzyme catalyzes that reaction, as in increases the rate and stuff. Now, all enzymes are pretty much proteins of some sort. And so that enzyme will naturally go cut something, right? So there'll be an enzyme which breaks down alcohol in your system and creates some of the compounds which give you a hangover, right? So now you can theoretically have a molecule that is shaped sort of like that alcohol molecule, which goes and sits there, but the enzyme cannot cut it.
Okay.
So you have deactivated that enzyme.
So are people working on this?
I'm sure they are, but there are no known hangover cures right now.
Okay.
Right, but this is the idea, right? So what people realized was with bacteria, the systems that make a bacteria run are fundamentally different from the systems that make a human run. Mm-hmm. So it's very easy to find out a key which goes and hits a lock in the bacteria, which touches nothing in the humans.
Right.
But the moment you change that to a virus, because I said the virus hides within and hijacks the human cells, much harder to go hit that virus. You have fewer locks, fewer targets to address. But this is the general idea that started, and a lot of new drugs have come from that idea, which means you need basic research on disease biology, and then you come up with these potential ideas. Okay, there is a target here. I think these drugs might be a target. And then you test that out in a test tube. Right? Now you can even create that enzyme or protein synthetically. You can get your drug, and then you create some sort of an assay, right? A test. Right. Something which says, okay, if the drug has worked, then you get a yellow color, or something.
Yeah.
And then you test it purely in test tube. You're not even talking about cells right now, right? Purely in the test tube. If that works reasonably well, if you have, say, 100 compounds or 1,000 compounds that work very well in a test tube, then you start testing it on individual cells, right?
Okay.
So you take a bacterial colony, you add some antibiotic to it, see if the colony is destroyed.
Genocide, basically.
Yeah, of bacteria.
Bacterial.
Well, criminal. So you see that, and then once you have that, then you start testing it in small animals, you test it extensively in computers, but you still can't get away from animals. I mean, because a mouse is still the best replicator of the human body that we have, right? Yeah. So you might start from 10,000 compounds, which might, be a cure for diabetes, work amazingly well in a test tube. By the time you even come to the first human test, 10,000 would have become 1.
Okay, right.
Right? So a lot of the time, this 10,000 to at least the 100, the work is done by basic science researchers, often on public dime. And This is the part we don't understand often in India, right? A lot of the time, it's academic research, which brings it to a level where the market forces can
can take it over.
Exactly.
And then, right.
Because after that, I mean, some of this work might also be done in parallel by pharma companies, but a lot of it is done by academic, non-profit, public-oriented research. But once it gets to a stage where, okay, you know what, this looks promising, maybe it has a chance of being a drug. That's when you have your clinical trials and that whole mess.
So you're saying till that 10,000 to 1 process is largely done by academics, and then the pharma companies take over?
It depends on where you start your 10 or 1,000. Usually what hits phase 1 clinical trials is itself in the hundreds or thousands, right? So you have, think of it as a funnel where till you get to anywhere near clinical trials, you have both private and public research. But typically once it gets to the clinical trial phase, you have a big pharmaceutical which is running it.
Sure.
Unless you have some PSU equivalent somewhere which is testing.
Yeah, let's not get there. Okay.
Right? So basically what you do is, once you have demonstrated that this is a drug that has potential, that is reasonably safe in animal models, etc., you take whatever you think is the dose of this, divide it by 1,000 and give it to healthy humans. Right?
Okay.
Give an extremely small dose, just see if anything happens. Right? And then you increase that dose a little bit, see if anything happens. So you're just testing the safety in phase 1.
Right.
Phase 2, and any safety red flags occur, you're out right there. Or the efficacy is not great, you're out right there. And then you get to phase 2. Phase 2, you start testing, how much of the drug actually reaches where it's supposed to, how much gets absorbed by the bloodstream, how you start testing effectiveness a little bit in a small population. And then once you have some reasonably positive results, you scale up for phase 3, where you want to test a large section of the population and see if the drug works for them. Right? So in this, again,
this is still clinical trials phase.
Yeah,
right? So you need
phase 1, 2, and 3. And even for getting phase 1 clinical trials, the drug regulator has to approve, saying, yes, you can do a phase 1.
Right.
Because you're giving this to humans now.
Humans,
right?
If something goes wrong, then of course
exactly. So there is the huge principal agent issues, information is asymmetric, you need a regulator, you need a Food and Drug Administration, you need a drug regulatory agency of some sort to say that, yes, I will allow you to go to the next stage.
And now people volunteer for such things when you do clinical trials with
I mean,
human
I
beings?
remember when I was in the US, within buses, there were these ads all the time saying, earn $1,000 from this 1 week stay at some place. And you would basically be enrolled in clinical trials and as a healthy human.
Yeah, slightly scary, but sure. I mean, I see the importance of it.
So in this, right? I mean, this is an interesting time to be talking about randomized clinical trials because a bunch of economists won a Nobel Prize for applying RCTs to economics, but their origins are here, right? So how do you know if your drug works? How do you test whether this works? You can't just say, okay, I gave this person medicine, he became better the next day.
It could be, again, that could be coincidence.
That goes back to homeopathy level.
Yeah,
I'm sorry. And homeopathy is junk science and not real research.
What are you saying, Pavan?
Yes,
Okay.
I'm going to be offending everyone. But that's the thing. You need to basically be able to find a robust system in order to test any of these drugs. And that's where a randomized double-blind gold standard control trial comes in, right? Where you want to give, in case where there is no other alternative, you want to give a sugar pill, which is a placebo, and your active compound, and you don't want either your patients or your doctors and nursing staff to know which is which.
Which is which, of course.
Because it can influence behavior.
Yeah.
So that's why it's double blind, right? So because you don't want, because anything suggestive has an effect. And the placebo effect is an amazing thing of, we talk about mind over matter and so on. The placebo effect is mind over matter.
It's real.
And I mean, it's been proved that an injection of saline water is more effective than a sugar pill at treating pain.
Right, okay.
So one placebo, because it's a more dramatic intervention, feels like it helps with pain more than another placebo, right?
So homeopathy is not even getting the placebo effect in its strongest dose.
Well,
I mean,
with homeopathic dilution, I think the placebo effect stays the same. Right? But importantly, I told you, right? Drugs have to enter our mouth, go to the stomach, get absorbed. Our body has 10 to the power 27 molecules, roughly.
Okay. So a few here and there,
So that's all. So when you take a 500 mg pill, you are taking about 10 to the power 20 or 21 molecules.
Okay.
So that is 10 followed by 21 zeros. You're taking that many molecules.
Right.
More than 90% will never pass through your intestines into your bloodstream. Less than 10% will get to your blood.
Really?
Most of the time. The digestion is that way, absorption, etc., etc. Of the 10% that gets in, they go everywhere. They don't know that, especially think of a painkiller, right? Because sometimes we have a different idea with, say, TB. It knows where lungs are, so the drugs will go there. It's not intelligent. It's a tiny, stupid molecule. It can't think. So it spreads everywhere in the blood at a certain concentration. Mm-hmm. At that concentration, it has to work in its designated area, which is why the fundamental principle of drugs is called the law of mass action.
Right.
The more you give the dose of that drug, the higher
the probability that they'll find the right place and then do its work.
And do their work, right? So what you study with drugs, two important things are toxicity and efficacy. So you have something called an LD50 and I think ID50, where the idea is at a certain concentration per weight of the animal, 50% of those animals, the infection will be cured. That's the effectiveness level. So you find out. So you do a dose response curve. So you give it 1 mg, give it 10 mg, give it 100 mg, see where you start seeing effect. And you find out that midpoint, where it's 50%. And then you do the same thing for lethality. What concentration does this kill half the mice?
Right.
And you do that. And you want your lethality to be orders of magnitude higher than your efficacy. The moment the two start coming close to each other, you get into dangerous territory.
Sure.
Heroin, for example, is something where the lethal dose may be 2x the effective dose.
Right.
LSD, in comparison, the lethal dose is more than 1,000 times or 10,000 times, or it just may not be lethal compared to the effective dose. Right. So the same thing works with medical drugs as well. Right. So this is something we need to care about, and this is why homeopathy, which just dilutes everything, it dilutes up to 10 to the power 400 times.
Yes. Yeah.
So the probability of finding one molecule in a swimming pool of your medicine is actually less than 1%.
So let us take a break at this point, and we'll get back and speak about a whole host of other interesting things, such as, are we developing resistance to antibiotics? Why are antibiotics so expensive? A lot of them at least, and a lot more other interesting things.
Thanks, Anupam.
This is the amazing story of something awesome. Once Chuck decided to start a podcast, and so he did. The end. Okay, that was a crappy story. But I've got some really cool stories over at my new show, The Origin of Things. On this podcast, I look at the stories of how brands came into being and sometimes evolved out of quite unexpected circumstances. And to make it really fun, I reveal the name of the brand and sometimes a category only at the very end. The show is 5 to 7 minutes per episode and perfect for trivia junkies and brand nerds, especially those with short attention spans. New episodes out every Wednesday on IVM Podcast app or website, or any podcast app or site that you happen to prefer. End of story, they lived happily ever after.
Welcome back to the Pragati Podcast. I have with me Pavan Srinath, and we are talking about medicines.
Hey, Anupam.
Hi, Pavan. So, we've kind of gone through until now about how antibiotics are produced, how are they discovered rather, and then how are they produced to the mass market, going through the clinical trials, probably getting approvals.
And this is the same for antibiotics and many small molecules for everything from heart disease to diabetes to other things, right?
Sure.
Whenever your solution is a standard chemical, this is broadly the route.
Sure.
Modern medical therapies are going to the next level. You have biologics, biosimilars. Let's stay away from that.
Yeah.
We'll stick to 20th century medicine.
Right.
Because that's challenging enough for India.
Which we still don't understand. So as we are kind of finding out now. So I hope your description of the complexity of the process is not an excuse as to why medicines are so expensive. Now, we find that in the US, in India, depending on different kinds of systems, but you find, especially in the US, there's been a big hue and cry of late that medicines are extremely expensive. Whatever generic medicines, I'm not talking about whether it's antibiotics, whether it's antiviral, whatever, right? Medicines are expensive. And so in India and in a lot of other places. Now, this also then results in some kind of pain for people who procure medicines and have to, at least are dependent on it for their survival. And then which obviously gains or gathers government attention, civic society attention, and then, there's a clamor for something needs to be done. We'll go into later about what that something is and so on. But why are medicines so expensive?
It's simply because the research that it takes to develop a potential new drug is extremely expensive. And once you have discovered a potential drug, the process of ensuring that it is safe for humans to use and consume, especially, there are many drugs that are chronic drugs as well, right? You don't just take them when you have an illness, but, if you're taking a acidity inhibitor, you're probably taking that for the rest of your life, right? So you need extreme safety standards, and we are paranoid about human safety. And then it has to be better than everything else that exists right now in the market, right? And for it to meet those conditions, it involves a lot of money. So the early research that I described to you, the basic sciences research, some of the stuff, that stuff is expensive on its own and it's paid for by various people. But just the process of getting something from an animal model all the way to phase 3 clinical trials and out into the market takes about $1 billion per drug.
Right. Wow. Okay.
Per drug that makes it out.
Right.
Not per drug that goes in.
Obviously.
So most of them don't go out.
So the cost of the successful drug also has with it the cost of the hundreds of unsuccessful ones.
Right? And then you have to prove that this new thing is better than everything else that exists. Now, I mean, imagine something like mental health medication, where, by the way, there's very little rational drug design. Okay,
Sure.
We don't know how most of these meds work.
Yeah.
We have ideas and theories. If you want to create, say, a new drug against depression, and you have a bunch of, and some have been made, and you have these bunch of drugs that exist, and you're even running a clinical trial is dicey. Can you prove that your new drug works better than all the other above? And then can you market it and sell it to doctors and get them to prescribe it?
And by how much?
And by how much?
Right? If you bought out a new drug and it's 2% better than the previous one,
Right.
but it's 10x more expensive, then no one will buy it.
No, in fact, it's along these lines that I'm not sure if it's Pfizer or Glaxo or SmithKline, a big pharmaceutical company completely shut its neurological disorders division in the last few years, right? After sinking in hundreds of millions of dollars of their own, they just decided, look, we are better off shuttering this entire business division because we don't think we're going to make any money in the near future. Right? So you can have nasty things happen, but you need that kind of money.
And
now,
then these are big pharma.
Yeah,
these are big pharma. Think about small. I mean, so is there no market for smaller players to actually do all of this? And
if you want to get through the regulatory process and get your drug sold to Americans and Europeans, answer is no.
No.
Okay, and you need serious money. The way to get around it, which is happening with our new therapies, the gene therapies that we've been hearing about, some of them are targeted against very rare diseases. Like you might only find 15 patients. What sample size are you going to do with 300, 400?
Right.
It's a rare disease, genetic disease. So that's become this regulatory gray area where, you know what, you're coming up with a credible cure for these 15 people who suffer from this thing. Why can't you test it? And however flawed your results are, let's go ahead and test it. Especially for diseases that are terminal, where, people don't have much hope and so on. Cancers and genetic diseases. So they're reducing the cost by attacking some of these problems. And that's the way in which new gene therapies are coming, where your unit costs are itself higher. Right. So your $1 billion might actually be 10 billion for a gene therapy. Mm-hmm. Right. So they're doing some things like that, which is good.
Let's get back to now your standard drug, which is out in the market. So we said that, the cost of that also has a cost of failures. Just to understand that, what would be the ratio of the success to failure? We spoke about until the clinical phase. You start off with whatever 10,000 or even 100,000 kind of different compounds, and one will make it to the point where it sounds promising, right? And then what is the ratio beyond that? So if you got, let's say, 10 promising...
So typically anything that passes a phase 3 clinical trial, you will try and get it into the market immediately. Right. That's the whole point of...
Okay.
Especially when you spend that kind of money on a phase 3 clinical trial. And that's why you have all these medical thrillers, right? The company has sunk in $800 million and then it fails or it shows a nasty side effect. And then there's a pressure to suppress or a pressure to publish favorable results. So there's a lot of evidence bias and some fantastic work done by Ben Goldacre and many others across the world to release all clinical trial data. Because, with sampling and bias, if you have done 10 studies on your drug, 4 were positive, 4 were neutral, 2 were negative. And then you delete the negative results and the neutral results, only show the positive. You can say that your drug works wonders.
Yeah.
And the reality is your drug may be no better than whatever already exists. Right? So there are all kinds of challenges like that. So big pharma, there's a reason why we should ask hard questions of big pharma, but this is the kind of money it takes. And now what happens is after this, the drug gets patented, right? Or in this process, the drug gets patented. And you typically now get a 20-year patent on most drugs.
20
right?
years.
And the number might vary across places, across different types of things. But so you have that time, which is your monopoly time to recover costs. Because the unit cost of your drug is nothing. It's like a few paise. Which is why once you have created a compound, it's very easy for an Indian pharmaceutical to find out what that compound is, find out a nice, inexpensive, robust way to create that thing. We have amazing synthetic chemists in the country, and then you can mass produce that with very little cost. Right? But you want to give a pharmaceutical company or whoever has the patent a monopoly for 20 years to actually have a chance at recuperating the kind of monies they have sunk in. Typically what people have done is they know that most of their rich patients are in the US or Europe. You also have insurance programs which are capable of paying for them. So drug companies expect to recover most of their costs in North America and Europe. And then eventually they'll introduce the drug in India while under patent as well at some cost. Sometimes we might consider this insanely expensive. Sometimes they might think it's significantly cheaper than elsewhere, all of that. And in the meantime, in India, a lot of times pharma companies start making them.
Of course.
Increasingly they can't, because we've signed up for global treaties. And once the patent expires, the drug is available for really, really cheap. And it also is important that you have multiple generic pharma companies which are making the same thing. You remember the controversy with this guy Martin Shkreli, right?
Yeah.
Who bought up a generics drugs company in the US and just hiked up price of one essential drug for a small section of people. So you want, again, competition, you want easy entry, you want anyone who knows how to do synthetic chemistry well, who knows how to do production well, to come and make your drug at high quality.
So let's again tie this back now with what we started. So we understand that there is, that there are generics in India, and we're doing, okay on that, though we have, I think increasingly we'll start fighting IP laws and IP battles.
Yes. No, and you know what? One of the interesting things that Trump has done is Trump basically asked a bunch of pharma companies, why are Americans footing the bill for these drugs more than anyone else?
Sure.
And honestly, it was one of the most legitimate questions Trump has asked.
Yes, that is legitimate, because they're spending billions and billions of dollars on R&D.
Right.
And the benefits are now distributed across the world without proper...
And Americans probably end up paying higher prices for a whole host of reasons.
Yeah, in fact, because there are generics and other countries are copying it, you have the patent which is only, let's say, viable in Europe and US.
Purchasing power as well, right?
Yeah.
Your insurance scheme has enough money to pay for something. If not insurance, Medicaid or Medicare, there are many states in which state governments can't negotiate with pharma companies on the cost of these drugs.
Right.
Because they're like a monopsony and a whole host of reasons. Whereas somebody like the National Health Service in the UK can at least do bulk contracts with pharmaceutical companies, saying that, look, we have 10,000 patients with this heart disease. I'll buy medication for all of them from you. And so, you reduce marketing cost, you make the whole thing cheaper. So you have all of that. So, and Indian generics have had a horrible track record in the last decade, especially in the last decade, where drug units meant for export to the US, FDA has a robust sort of, they check the entire supply chain and do tests at every level. And they check tests. And if you have computer files of sort of test data, they check whether you've messed with the file data. And they know how to do all of that. And one of the things they realized, a couple of big pharma companies got into trouble because their quality control labs were terrible. So your drug is fine, but you don't know how purely you've made the compound. You don't know whether it is effective. And which is why, and this is the stuff that is being exported to the US. Domestically in India, our drug regulator effectively, if you get a drug passed in the US or UK or some European drug authority,
you get a green tick mark here.
Basically submit the same thing over here, and they'll say, okay, it's been approved elsewhere.
If FDA approves it, you get our this thing as well.
That's it.
Yeah.
Beyond that, what safety analysis is done is, once in a while, our drug controllers, the drug regulators, basically go to pharmaceutical stores and buy medicine. And then they check various purity and other terms. Random. Whereas what FDA does is it looks at each batch and it tests it at every site, so that once you find a failure, you can trace the point of origin of that failure, and you can, withdraw what medicines you want, cancel what you want. You have actionable insights. What we do is very, very, very limited.
That's still on the drug regulation side. I'm still interested in the development of drugs.
Right.
And I want to close the loop that we started with, which is, it seems now that we are heavily reliant on drugs which are made in the US. And then we can copy them at some level, not copy them, or do something about it. However, of course, as you mentioned yourself, we do not have the same level or the same kind of diseases in India as there are in the US. There might be some which are overlapping, of course, which again can be, you can then use the medicines in the US. But there are a lot of diseases which are unique to India. And similarly, I would imagine they are unique to different countries in Africa, unique to China, unique to this thing, and so on. So how do we then develop the drugs which are unique to India? Or what is the solution ahead for those things? Now, you can't rely on our current set of companies which are heavily reliant on generics to do this kind of research. I don't know if they have the money, if they have the expertise, and so on. Our government regulations, given that our IP laws are so weak, doesn't really seem to encourage new research and development of of drugs. And then of course we do stupid things which I do not really want to get into, that we put price caps. So let's say you have a rare case where an Indian company comes up with a new drug, and then we put a price cap on it and make sure they are never profitable. How will India ever produce drugs for Indian diseases is my big question, and I think we'll end it with that.
And it remains a big question, but a few points of thought there. One, there have been a few pharma companies in India which have started doing this kind of research to develop new lead compounds for in the drug discovery pipeline, right? So that they go down this funnel and eventually get to clinical trials. The few who have done it, the moment they have found out a promising compound, have sold it to a big pharma company, because the amount of money it takes to get through clinical trials is heavy.
Right.
And no Indian entrepreneur has decided, okay, I will get into the drug development game and spend that kind of money. Very few can. A notable exception is Biocon, which is actually filing for new patents. They're doing biosimilars, so they're doing different drugs as well that are beyond small molecules. Insulin itself is a peptide, right? It's a bigger molecule, and you can think of things bigger than insulin.
Sure.
So with the exception of that, the one or two cases in which we've developed new potential things. So in that sense, those few companies were making pharma, again, for the world or for the West.
Right.
Not for India. And a lot of the times we'll hear news about, IIT Kanpur researcher discovers new drug for malaria.
Right.
All they've done is they've published a paper which has proved that there is this new thing which has some promise. They've not even gotten to a promising lead compound level. Right? But that is declared as triumph. And then the news vanishes. Because what they did was good work, but it was very, very poorly reported, right? So they did great stuff, but then what happens after?
Yeah.
So the first thing in this is we are not spending enough on the public layer of all of this, which markets cannot do a good job at. We don't do enough basic sciences research. We don't do enough drug-related research. My estimation is Government of India funded drug-related research projects don't cross 200 crores a year.
That is peanuts.
The entire Department of Biotech research budget is 2,000 crores. So the universe of biology and biotech.
Right.
And the drug-related stuff will be less than 200 crores. So you're not going to get anything from there, right? So the first is that number needs to be dramatically ramped up. And when we do mission mode stuff, right? Okay, we'll do mission mode on dengue. What are you doing on dengue research? Right? What are you doing on doing various actual studies? Not much, right? What is even preclinical? How are you studying the virus? How are you studying, various vaccine opportunities? Not enough, right? So that is just missing. So until that comes into place, everything else is a band-aid. Okay. Second, along with that, you can have philanthropic money that goes in and all of that. Gates Foundation has honestly helped a lot with HIV and other stuff in India. And they've pumped in a lot of money. My guess is Gates Foundation at one point would have spent the same on drug development related to India same or more than the government of India, right? On drug development. Okay, one foundation alone. The second layer is, like you said, what is the incentive for anyone who's making a drug that is meant for Indians?
Yeah.
The correct way for the government to approach this would be to say, okay, I realize that something like malaria or dengue is a public health challenge. Therefore, I will buy 100 million doses of this from you.
Exactly.
Let's work out a deal.
Yes. Yeah.
So then the government pays. What the government wants to do right now is to say, look, this is essential drug. So I'll just decide what the price of this thing is so that it's affordable. And then you just make it. So they want the company to pay, right, to the point of ruin. In which case, you have created a business environment where nobody wants to create drugs.
Of course. I mean, that it makes no sense
So
whatsoever.
that is the big reason why we should make those decisions and uphold patents. Because if you want to create drug companies that are creating drugs for India, you need to send clear signals that you will respect their IP and you will allow them to charge, accordingly.
Yeah, but so which means just going by your thread of logic, we need to start taking public health seriously, increase our expenditure on public health at various levels. One, we could fund R&D ourselves. Two, as you said,
no, and it's all of the above, right?
Yeah.
And I'm talking here purely about basic research, and we just need a lot more funding on it.
right?
And so that's something we need to do right now, because drugs do take 10 to 30 years to get from bench to market. Yeah. So let's start the bench stuff now.
At least now.
And we can take 10 years to figure out what kind of regulatory systems we need to enable this kind of an environment, right? Having said that, our patent laws, however they stand now, I'm by and large in favor of them. I think the Supreme Court and others have interpreted it very well. This old case with Gleevec.
Yeah.
TLDR of what happened back then. Gleevec is this compound called imatinib. It was a new generation drug against glioblastomas, brain tumors. And it was much better than the old world chemo, great. But it was a drug that fell in this, gap period between India having old patent laws and adopting and sort of signing up for global treaties.
Sure.
So in that gap, it sort of missed through. And then what the guys who made imatinib did was they added a salt to it. They said imatinib mesylate is absorbed X times better in the bloodstream, is more bioavailable, and is therefore a better drug. This is a significant innovation. Give us a new patent.
Yeah.
And they got it. But Indian Supreme Court said, Sorry. No. And as someone who worked in a lab, I can tell you that changing the salt on a compound is not that complicated.
And it's not a big
innovation.
innovation.
So it's basically something that was purposefully left out. So it was no specified salt in the original patent, and it was not sufficient innovation to deserve a new patent. This process is called evergreening, and it's been talked about a lot.
Yeah.
And to that extent, I'm all for how the Indian regulators and courts have looked at this, because Big Pharma tries to do too much. They want to increase their monopoly regime beyond the legitimate amount, and that's not kosher. But that to me is good. The bigger problem is this business of essential drugs and how the government treats anything which it thinks is expensive and is essential for the public.
You've got around 8,000 odd drugs in this national list of essential medicine. It could be actually far more than that, but I'm not sure. And anything within this national list of essential medicine has some form of regulation on how much it can be priced and so on. And according to me, that kills the market. You're not going to have private players spending a lot of money and trying to build new drugs as long as you have this kind of crazy stuff by the government. I understand the need for it, that of course, ultimately affordability. I mean, you can't have people dying because of lack of affordability. That's a public health, and I mean, by and large, also policy decision that we have to undertake.
Right. But
this is not the right way to do it.
Yeah, and in general, yes, if India currently disregards intellectual property, it can benefit significantly. If we just waive off all foreign treaties, I mean, they have other repercussions, and just shamelessly start taking all drugs under patents and making them and producing them for the world.
Right.
It'll be great for Indians. but it will mean that no new drugs will be developed for Indians in the foreseeable future.
So as always, with everything in public policy, you have to find that right balance between these various forces.
And I think the no-nonsense thing is increase public spending on research.
Yeah, I think that's a given, because by a lot of the stories that we've heard today and just understanding how this works, developing a new drug seems to be a story of serendipity. A lot of luck is involved, but you have to make that luck happen. And you can only make that luck happen by doing a lot of trials around it.
We are now at a level where it need not be a blind search in the dark.
Right.
It can be an informed, open-eyed search.
Yeah.
In mysterious places.
Yeah.
Right? So we are at a place where we can do this while luck plays a role. We can actually get there with resources. I think biology is advanced enough for us to do that. And now it's about our political will, as you would call it, to actually get us there.
Right. On that, I'm not sure. Is it a happy note? Is it a depressing note? I'm not sure. I'll let our listeners decide. But thank you so much for joining in. I learned a lot during this particular
Thank you, Anupam.
episode.
Thank you for indulging me and hosting me on this episode. Thank you so much.
Cheers. Thanks.
Thank you for staying with us till the end. If you have any questions or comments, do write in to podcast@thinkpragati.com. And hey, if you like the podcast and listen to us on iTunes or Apple Podcasts, please leave us a rating and a review. It'll mean a lot to us. The Pragati Podcast is available on the IVM Podcast app and pretty much every other podcast app and platform. We are there everywhere.
Filter coffee is a fascinating beverage. You need to pick the right beans, blend them in the right proportion, roast them to perfection, and slow brew at the right temperature to get the perfect cup. Which is exactly like great conversations as well. You need to track down the most interesting minds, get them into their zone, and settle down for an unhurried, unscripted chat. And coffee for me is always, always, always best enjoyed with friends. I'm Karthik Nagarajan, and do share my table as I meet some of the most interesting people I know and sit them down for a strong cup of coffee and an even stronger conversation. Join me every Wednesday for a freshly brewed episode. This is not frappe. This is the Filter Coffee Podcast.
Advertising is dead. Yep, you heard me right. Advertising is dead. We're all in the content business now. Let's not call it news, TV, radio, etc., etc. It's all content, and we're in the middle of this weirdly exciting phase where all the borders and lines that have been drawn over decades has been swept away by this lovely thing called the internet. We're a show where we don't dwell on just the stuff that is now, but rather the wider stuff about advertising, media, content, and the whole goddamn circus surrounding it. Tune in every Tuesday for our weekly unboxing of the mystery box we used to call advertising. I'm Varun Dugirala, co-founder and content chief at The Glitch, and this is my new podcast, Advertising is Dead.